So quickly, a little bit about Dr. Tremblay before we get started. Professor Jacques Tremblay obtained a PhD in neuroscience from the University of California in San Diego. He has been working on the development of cell and gene therapies for hereditary disease since 1987. He's published hundreds of articles and is very committed to the clinical application of his research work. He received the award for Best Researcher in Quebec and the award for Best Researcher in Canada, presented by the Muscular Dystrophy Canada for his work on DMD. He is currently using CRISPR-Cas9 and the prime editing technology to correct mutations in a number of neurodegenerative disease-associated genes, and he is currently trying to identify the best delivery method to deliver the components of these gene editing technologies. So thank you everyone for joining us, and we'll go ahead and start Dr. Tremblay's lecture.
The title of my conference is Development of a Gene Therapy Approach for Friedreich Ataxia by Deletion of the GAA expansion in the Frataxin gene. Friedreich Ataxia is a neurological disease which is due to an autosomal recessive mutation. The prevalence of this disease is about 1 in 20,000 to 1 in 50,000. In the United States, it is estimated that 10,000 persons are affected by Friedreich ataxia, and in Canada, this would be about 1,000 persons. The main symptoms of Friedreich ataxia are movement coordination problem, cardiomyopathy in about 59% of the dead, and diabetes in about 10% of the patient. The most frequent mutation responsible for Friedreich ataxia is the insertion of a long trinucleotide repeat, 60 to 1,700 GAA repeat in intron 1 of the Frataxin gene. There are also some rare mutation of a single amino acid in several different positions of the Frataxin gene. Death frequently occurs between 16 and 30 years of age. The age of death is inversely related to the length of GAA repeat. With longer GAA repeat length, the death occurs earlier. The presence of the long GAA repeat induces the formation of what is called as R loop. This is an interaction between the messenger RNA and the DNA, and it also increases methylation. The formation of these R loops reduces the formation of the frataxin messenger RNA. The frataxin protein, which is formed from the messenger RNA of frataxin, is involved in the sulfur iron metabolism. There are several treatments for Friedreich ataxia which are under investigation. Unfortunately, most of these approaches do not lead to a permanent cure. The hypothesis underlying my research is that excision of the GAA repeat by the CRISPR-Cas9 system will increase the expression of the frataxin messenger RNA. The CRISPR-Cas9 system was discovered in 2012. It used a Cas9 nuclease, which first starts its interaction with the DNA at a site where there is a NJJ nucleotide sequence, N being NA nucleotide and J being 2-guanine. This starts the interaction between the Cas9 nuclease and the DNA, and the precise site where a cut will occur is determined by the presence of a single guide RNA, in yellow in this figure, and there is a sequence of 20 nucleotides in pink that will be complementary to a sequence of 20 nucleotides in the DNA. When this complex is formed, there will be a double-strand break that will be produced at exactly 3 nucleotides from the NJJ sequence. When a double-strand break is produced, this will be repaired by homology-directed repair or by non-homology and joining. By inducing DNA cuts at 2 sites within the frataxin intron 1, this will permit to remove the GAA repeat, which is responsible for the reduction of the production of the messenger RNA of frataxin and thus the reduction of the frataxin protein. We have initially tested various pairs of SP-Cas9 single guide RNA to induce cuts at different positions in intron 1 of the frataxin gene. When the adequate pair of single guide RNA is identified, this permits to cut and remove the GAA repeat, and this produces a band of DNA that migrates further down in the gel because the migration is related to the length of the fragment of DNA. For the in vivo treatment, the Cas9 gene and the sequence of the 2 single guide RNA have to be delivered by an AAV to the mouse model. Unfortunately, the AAV virus can deliver a maximum of 4,700 nucleotides. Unfortunately, the SP-Cas9 gene by itself is 3,999 base pair, and it's too big to be delivered with the other component, the 2 single guide RNA, by a single AAV. Fortunately, there are various Cas9 nucleases that have been identified in different bacteria, and one of the smallest one is from Campylobacter jejuni, called CG-Cas9, and it's smaller than the original SP-Cas9. The adeno-associated virus can deliver its content more or less effectively to different organs, and this depends on the protein which are at the surface of the AAV. A promoter is a sequence of nucleotides that control the expression of the gene which follow that promoter. There are different sizes of promoter, mini CMV, CMV, CBH, and EF1 that we have investigated, and we have selected the CMV promoter because it's a powerful promoter and yet it is in a size which is adequate to permit delivery of the CG-Cas9 gene by a single AAV, including also the 2 single guide RNA. We have tested the expression of the CG-Cas9 nuclease under the control of 4 different promoters in HAK293 T cells and in YJ8SR cells, and we have found that the CMV promoter is the strongest. The CG-Cas9 nuclease can thus be delivered by a single AAV under the CMV promoter, and the AAV will also contain the 2 single guide RNA. For delivery to the heart, we are using an AAV9, and for delivery to the brain, we are using AAV PHP.B. There are 4 different mouse models which are commonly used to study Friedreich ataxia. The first one is the Kiki mouse model. The second one is the Kiko mouse model. The third one is the YG8SR mouse model. And the fourth one, more recent, is the YG8SR 800 mouse model. In fact, we have used mostly the last 2 mouse models, and the YG8SR contains about 200 GAA repeat, whereas the newest one, the YG8 800, contains 800 GAA repeat. we have documented that both male and female YG800 mouse do not develop as rapidly as normal mouse. The concentration of the frataxin protein is very low in various tissue of this mouse model. Several parameters of the heart are abnormal in this new mouse model. The left ventricle volume is higher in the new mouse model. The ejection fraction is lower, and the left ventricular and diastolic wall thickness is also lower in this mouse model. We have found that the new mouse model takes more time to cross a notch beam or a T-inverted beam, and it makes more foot error in both the notch beam and the inverted beam. We have also observed that the female YG800 remain hanging much shortly than the normal mouse. Following an intravenous injection, the AAV9 is detected very well in the liver and the heart of the YG8SR and YG800 mouse model. It is also detected to a lesser extent in muscle, brain, and cerebellum. We have tested 5 different single-guide RNA which are able to induce a cut in the DNA in intron 1 before the GAA repeat, and 5 different single-guide RNA which are able to cut in intron 1 after the GAA repeat. We have then tested these single-guide RNA in pairs, one cutting before the GAA repeat and one cutting after the GAA repeat, to identify the best pair of single-guide RNA to delete the GAA repeat. We have found that the best pair of single-guide RNA to remove the GAA repeat is the single-guide RNA U4 that cuts before the GAA repeat, and the single-guide RNA D1 which cuts after the GAA repeat. This removes the GAA repeat fragment, and this permits to increase the messenger RNA of the frataxin, and this also increases the expression of the frataxin protein in the cells that have this deletion. We then use an AAV9 to deliver the CGCAS9 gene and the sequence coding for 2 single-guide RNA to the heart of the YG8SR and the YG800 mouse model, and in both cases we were able to detect a low percentage of deletion of the GAA repeat. Although the deletion of the GAA repeat was low, only 1 to 2%, we were able to detect an increase of the messenger RNA of frataxin in the heart of the YG800 mouse model. Following an intravenous injection of AAV9 coding for CGCAS9 and the 2-guide RNA permitting to delete the GAA repeat, there is a better growth of the YG8SR and the YG800 compared to mice treated only with saline. The single-guide RNA that we use to induce cuts before and after the GAA repeat may also sometimes induce cuts at other sites in the genome. These are called off-target cuts. This was investigated using the BenchLink website. For the single-guide RNA U4, only one annotated gene was identified, but it had only a very low off-target score. For the single guide RNA D1, this one targeted a pseudogene, that is an inactive gene. The sustained expression of the Cas9 nuclease may induce some problems. For example, there might be antibody developed by the host against this foreign protein. The sustained expression may also increase the risk of off-target mutation. To avoid these problems, we have therefore investigated if we can control the expression of the CG-Cas9. One method to control the expression of CG-Cas9 is the molecular harakiri method. For this method, in addition to delivering the CG-Cas9 gene and 2 single guide RNA targeting the intron 1 of the Frataxin gene, we are also delivering 2 single guide RNA which are targeting the CG-Cas9 gene itself. Therefore, these 2 single guide RNA cuts a part of the CG-Cas9 and make this enzyme not functional. We have indeed observed that this harakiri method induces a cut of the CG-Cas9 gene and a reduced expression of the CG-Cas9 protein. Another method to control the expression of the CG-Cas9 nuclease is to introduce a premature termination codon within the CG-Cas9 gene. When there is no treatment, this premature termination codon prevents the synthesis of the Cas9 protein. However, when the cells are treated with an aminoglycoside, such as J418, this promotes the read-through of the premature termination codon, and the CG-Cas9 protein is then synthesized. We have then tested the effect of this introduction of a premature termination codon in both HEK293 T cells and in YG8-SR cells. As you can see on the left side, when there is no premature termination codon in HEK293 T cells, there is a deletion of about 19% of the GAA repeat. However, when there is a premature termination codon and no 418 drug is administered, there is absolutely no deletion of the GAA repeat. However, when increasing dose of J418 drug are given, there is progressively deletion of a higher percentage of GAA repeat with increasing dose of J418 drug. The same observation is also made in the YG8-SR cells. Ding et al. Have reported in 2019 that it is possible to increase the efficacy of the single guide RNA to induce a DNA cut by changing only 2 nucleotides within the constant part of the single guide RNA. This is called the scaffold. So we observe indeed that using scaffold 1 and scaffold 2 having the modification suggested by Ding et al., that we obtain a higher percentage of cuts of the DNA and therefore a higher deletion of the GAA repeat when using scaffold number 2. Following the delivery of the CRISPR-Cas9 components, we have been able to obtain a deletion of the GAA repeat and an increase of the Frataxin messenger RNA and protein in the heart of the YG8-SR800 model. We have also been able to control the expression of Cas9 using a molecular harakiri method or using a premature stop codon. In future experiments, In our future experiments, we would like to study the improvement of heart function of the YG800 mouse treated with an AAV9 coding for the CG-Cas9 under a CMV promoter, and also coding for 2 single guide RNA targeting the Frataxin gene in intron 1 before and after the GAA repeat. We want to also study the improvement of coordination of the mice crossing beams. We want to see if they can cross faster and doing fewer foot mistakes. We also want to study the improvement that will be produced by an AAV PHP.B, a different serotype, controlling the expression of the CG-Cas9 under a different promoter, the HSYN promoter, and also containing the 2 single guide RNA. And if necessary for improving the brain function, we will also study stereotactic administration. This research project on Friedreich ataxia has been supported by grant from the Canadian Institute of Health Research, by the T-Cell Network, the Cerveaux Research Center, the Programme canadien des bourses de la francophonie, from National Ataxia Foundation, from Ataxia Canada, and from AFM Téléthon. Oops, we cannot hear you. Okay.
Sorry. Thank you so much, Dr. Tremblay, for that presentation and for your really fantastic work in this area. Just a reminder to those in the audience, we have lots of times for questions. So if you have any questions for Dr. Tremblay, you can enter those into the Q&A feature, or you can raise your hand and request to go live and ask a question. So we do have our first question in, and that is, do you think the GAA repeat in wild-type frataxin is playing any functional role, even though it's in an intron? In other words, is there any evidence that deletion of the GAA repeat could affect frataxin transcript processing or frataxin function?
Well, at this point, we don't have any evidence that the GAA repeat in intron 1 has some role. But on the other hand, it's possible that some of the other sequence within intron 1, which are not the GAA repeat, may have influence on the expression of the frataxin gene. So that's why it's important to select where we're going to cut exactly before the GAA repeat and after the GAA repeat. We would like to remove as little as possible the sequence in the intron, because they may have some role.
And I apologize if I missed this during the presentation, but can you talk about the efficiency of proper editing and how that might translate into an actual in-human, in-brain treatment?
Yeah, that's indeed the very difficulty. We have to, the efficacy will depend on which serotype of AAVs we are using. The serotype is just by the protein which are on the surface of the AAV, they determine what kind of cell will be infected by the AAV, and they also determine how effective is going to be the infection. The dose of AAV also is to be used. The higher the dose, the more effective it will be. This is one of our complications, because the higher the dose, the most expensive are the experiments also. And the single guide RNA, again, choosing the best one to obtain a higher efficacy. So at this point, showing in animal model that we can obtain a good enough efficacy to really improve the symptom of the mouse model is what is the priority. And unfortunately, I must say that at this point, we are not currently working on Friedreich ataxia, because unfortunately, we have no grant to work on Friedreich ataxia.
Mm. Yeah, I was going to ask you what the next steps are with this process, with this project. But
the next step is to obtain a grant.
Are you actively applying for grants in the ataxia space?
Well, recently I applied for a grant that was for Ataxia Canada in collaboration with Genome Quebec, and although you know, only 3 grants were accorded, and none of them were on Friedreich ataxia. So this was very disappointing because most of the patients that are part of Ataxia Canada are patients affected by Friedreich ataxia, and yet the committee that selected the grants selected among the 3 grants one disease that is affecting only 1 person out of 700,000 around the world. Very, very rare disease. So this is one of the grants that was given, but no grants on Friedreich ataxia was given. So at this point, we are preparing for submitting grants to other agency. One of the complications in submitting grants is you're not supposed to submit the same project at 2 different places at the same time. So unfortunately, I had really big hopes that I would obtain the grant from Ataxia Canada because I've been supported for many years by Ataxia Canada. And in fact, they made this arrangement with Genome Quebec, which is a para-governmental organization, has lots of money. So Ataxia Canada thought they would double the amount of money that they had for research. But unfortunately, they were not forming the committee that was selecting the grants. And the committee that selected the grants did not select projects that were on Friedreich ataxia.
We've worked closely with Ataxia Canada for many years on a number of different partnerships. I know just overall, there's a great need for more and more funding going into the ataxia space because of all the great researchers and just the magnitude of diseases out there. I know, I think we're up to 300 different genetic forms of ataxia.
Identifying new genetics forms of ataxia is important, but on the other hand, there are many ataxia with genetic disease has been identified. And we have to work on treatment and not only identifying new mutation responsible for other forms of ataxia. Let's cure the ones we have.
So back to the delivery method. I'm curious, is AAV9 being currently used in any human clinical trials at this time?
Yeah, it's one of the grants which is used. The AAV-PHP.B was also a new a serotype, which is very promising. And in my lab, we are currently working on developing new AAVs for delivering to the heart and delivering to the brain, because this is really one of the limiting steps. We want AAVs that are more effective and more specific for these organs. So this is one ongoing research, and this is not only for Friedreich ataxia. Therefore, it's an ongoing project because we have other source of money for that.
So I think in the last couple of years, we just discovered a new highly prevalent GAA expansion disorder, SCA27B. So we have a question from the audience in that, do you expect that this same approach, since you're targeting the excising of the GAA repeat, could be used for a disease like SCA27B, which is similarly caused by a GAA?
Yeah, I don't know about that gene, but it is important that the sequence, which is a trinucleotide repeat, be within an intron because it's a non-coding region. For Huntington, for example, there is also a trinucleotide repeat, but it is within a coding region. And so it's producing an abnormal protein. We could aim at removing that trinucleotide repeat, but then we don't have as much choice of where we're going to cut before and after the trinucleotide repeat, because this isn't a sequence that codes for the protein. So we cannot remove it. So that's a complication. So I don't know about the other gene you were mentioning, but again, it's good to remove trinucleotide repeat, which are fortunately in introns and not in coding regions.
Yeah. Well, in SCA27B, it is an intronic mutation. Okay, so potentially it could be used in that same way. We have a question about what is the biggest concerns using CRISPR technology, and in particular, what are the biggest concerns for off targets? What could be possible repercussions of a off-target editing?
Yeah, well, off-target are due to the fact that sometimes CRISPR can cut in other genes than the place we want to cut. And these off-target, they can be predicted by computer, because we have the sequence of the human genome, we have the sequence of the mouse genome, and so we can find what are the possible off-target sites. and they can be identified by the computer, but they can be confirmed by experiments to verify if indeed there is a cut in this off target. So I think that by selecting the adequate single guide RNA, we can avoid cutting near genes which are important for the cell survival, or genes that could be involved in cancer, for example. We don't want to touch those. So it's possible to select adequate single guide RNA that will not produce problem.
I'm curious, did you also look in the, I know FA is also very much a cardiac disorder. How active was the CRISPR activity in the heart versus the brain?
Yeah, in fact, we did not do many experiments so far in a live animal model, but one of the problem with live mouse model is that sometimes the disease is not severe enough. And this was the case for the YG8SR mouse model that was initially developed by Dr. Fuchs. That mouse model had only 200 trinucleotide repeat. And the symptom in this mouse, although there is symptom, it's not always as good and as severe as we would like. And so when you do experiments in a mouse like that, if the symptoms are not severe, it's difficult to see if you have an improvement. And so that's why we were very happy to find that the new mouse model, which was developed by Jacques laboratory, it has 800 GAA repeat. And the expression of Frataxin is down to about 5% level in some of the tissue. The symptoms are more clear. And so if we do experiments to remove the trinucleotide repeat, we hope that the improvement of the symptom will be more clear. And so that's why we published the results of that paper. We were the first one to describe the symptoms in this new mouse model. The symptoms are severe. The amount of frataxin is low. And so if we do in experiments, we hope that we will be better able to show the improvement. And this is what's important to eventually request that the clinical trial be done. We have to show first in animal model that there is a good improvement.
So I know there are several ongoing and recently completed clinical trials using AAV in some way in Friedreich's ataxia. And we also had very recently a couple CRISPR technologies be FDA approved for the treatment of sickle cell anemia. I'm curious, I know it's almost impossible to predict timelines, but when might you expect CRISPR technology with AAV delivery to be a possibility for human clinical trials? What's the typical timeline look like in perspective?
The timeline is always difficult to predict because of when the grants are coming or not.
Mm-hmm.
Unfortunately, sometimes the projects are delayed because there is no grants. And for example, just in the field of microdystrophin, there is currently a clinical trial ongoing with microdystrophin being delivered by AAV. But researchers have been working on microdystrophin for more than 20 years.
Yeah, research can happen very, very slowly and then very quickly.
I always say to people, science never go back. We move forward slowly. Sometimes you do something, it doesn't work. It doesn't mean that you're going back. It doesn't work that way. So we are always moving forward. The progress forward depend on sometimes the amount of student you have to work on a project. And so most of the research in my lab is done by a graduate student. And I cannot place a graduate student to work on a project for which I don't have research money, because, I have to pay this graduate student. I have to pay him a tuition, to pay, a studentship. And I have to supply the reagents, and I have to make sure that there is a funding for 2 or 3 years for the project so that I can put a new graduate student on the project.
Well, I would just mention that NAF is about to open our letters of intent period for our 2025 grant. So perhaps we'll be involved in your work moving forward.
For those that are listening to this conference, the rate of success is unfortunately very low for grant application. For example, in Canada, the grants are coming from the Canadian Institute of Health Research. The rates of success is perhaps at best 8%. And when you have already one grant from one subject, the chance of getting a second grant in Canada from the Canadian Institute of Health Research is very low. And when applying to agencies like National Ataxia Foundation or Ataxia Canada, again, the chance of obtaining the grant are unfortunately very low. That's part of life of a researcher.
Yeah.
We are
Yeah.
researchers.
Oh, sorry, I didn't interrupt you.
I was going to say we are researchers of money, unfortunately.
Yeah, for us, within a year, we went from 80 letters of intent to almost 160. So we're, yeah, even with the NAF, the number of applicants for our grants is greatly expanding, which is wonderful. It means a lot of people are working in this area, but it also creates more competition and doesn't allow us to support as high a percentage as we have in the past.
Yeah, and that means that a lot of researchers are spending time writing grants.
Yeah. Oh, so sorry. Did you want to
I
finish?
was going to say, unfortunately, from one foundation to another, the grants format are different. The forms, you have not the same number of words for the summary or explaining the objective and all of that. So although you are going to submit a proposal, which in basic is very similar, because of the time to fill the form, you're losing time applying for different grants at different agencies.
There's a question from the audience that is kind of related to whether you anticipate the future of FA treatment to be one versus another, or the use of combinatory treatments.
Well, when giving
you imagine GAAr and CRISPR, for example, being used in combination within the same patient?
There's a new technology derived from CRISPR, which is called prime editing, that we are working on in our lab. Quite frankly, it's a fantastic technology. It permits to replace any nucleotide, and I remind the audience that the human genomes, you're receiving 3,200,000,000 nucleotide from your father and the same number from your mother. It's a huge amount of nucleotide. And yet with this technology, you are able to replace one nucleotide, the one you're targeting, you can replace it by the nucleotide that you want. So it's a fantastic technology to correct genes. We have already corrected genes in several different genes responsible for Duchenne muscular dystrophy or dysferlin or so it's working very well. The main problem is delivery. We have to deliver, but and during the conference, I was reminded myself that 70% of the Friedreich ataxia patient have a deletion, have a trinucleotide repeat, but about 30% of the Friedreich patient have point mutation, and these point mutation could be corrected by prime editing, which, as I find you, very fantastic. It works to correct all the different point mutation we have targeted so far. We've been able to correct them in cells and culture. Now the problem is correct them effectively in a high enough percentage of cell in a mouse model. And so I always say to my student, there is 3 problems in gene therapy: delivery, delivery, delivery. We have to deliver the therapeutic agents into as many cells as possible to have a beneficial effect.
So I'm not sure how familiar you are with GAAr, which is the NERV2 activator that was FDA approved last year for the treatment of FA. I'm wondering if you could imagine that specific agent. Would that interfere with CRISPR technology, or could those be used in conjunction?
I'm sorry, I missed the beginning of your question about what agent was supported.
GAAr or omaveloxolone. It's the only FDA-approved treatment for FA or any ataxia.
I don't expect that it will interfere with gene therapy. In fact, they may both of them may complement each other. So if you have a 10% improvement with the drug and you have another 10% which is due to gene therapy, hopefully the 2 effects will be cumulative and not contradicting each other.
Thank you. We had a question from the audience, which is, do you also work with other types of ataxia in addition to FA in your lab?
Not at this point. I have in the past applied for other type of ataxia, but again, no success. But I'm working with several muscular dystrophies, including Duchenne muscular dystrophy, dysferlinopathy, and another one, congenital myopathy, which is due to mutation in a gene called R1 gene. So in all of those different hereditary disease, we're currently working with the prime editing technology. We are able to correct the genes in cells and culture. The next step is to correct this in the gene in enough cells in vivo in a mouse model to really be able to go to clinic.
Great. So I think we're going to be wrapping up here in a minute, but just a last question. We have quite a few early career investigators on the call. I was just wondering if you had any advice that you give to a young up-and-coming scientist who's entering the ataxia space.
Be patient, you know, research is always, sometimes you get the grant, sometimes you don't, and you have to remain in the field. You have to keep applying so that your team will survive. And my team has been surviving for, I would say, 50 years now. So you know, you always have to make research and apply for different grants on different disease, so that you know, the worst thing that can happen to a researcher is to be with 1 year without having any grants, because it means that you are going to lose your graduate student, you're going to lose your research assistant, and you're done. So, you know, and the only way of avoiding having a complete absence of grant is to make several different grant applications all the time, writing grants all the time, applying for grants. And when it is submitted, go for another one. That's the only way to survive in science.
All right. Well, thank you so much for that advice and for your wonderful presentation today. On behalf of patients, thank you for your dedication and progress that you've made towards developing treatments for ataxia. Just thank you so much. Bye-bye.
Great
rest of your week.